Lecture 8, Integumentary System Part 2, Accessory Structures. We finished with the last lecture set looking at the integumentary system. Now we'll go through some of the accessory structures in this short lecture series. The integumentary system, remember, is divided into the cutaneous membrane, the epidermis, and the dermis, whereas the accessory structures we'll be looking at will be the hair follicles, exocrine glands, and the nails. Hair is found all over the surface of the human body, as over 95% of it is not on the head. Hairs are acellular, non-living structures comprised of keratin and formed in special organs called hair follicles that we can see here. The follicle is going to be deeper down into the overall hair root. There's the root plexus that has nerves that attach to the overall connective tissue sheath, and an erector pilae, smooth muscle tissue, that will bind that to the integument itself. From there, the hair root will extend up into the hair shaft as it pierces through the skin. There are sebaceous glands attached to the hair itself, and the sebaceous glands actually secrete oil or sebum. Here, deeper down into the hair, we can see the root and the hair matrix as it extends down. The hair papillae is simply going to be where blood vessels extend to the root of the hair to allow stem cells to grow and the hair to increase in length overall. Some of the functions of hair are thermoregulation, protecting against UV light, guarding the nostrils, ears, and eyes from foreign particles and insects, and then social function for mate, attraction, aggression, etc. Hair color itself is dependent upon the structure of the hair, thick, thin, etc., and variations in pigments produced by the melanocytes in the hair matrix. There are different forms of melanin that can cause dark brown or light brown, red, etc. Hair is mostly determined by genetics, but hormonal and environmental factors can also change hair color and condition. Hair growth and shed in hair growth is done in a cycle. There's an active phase and a resting phase. The active phase lasts 2 to 5 years, and hair growth grows at a rate of 0.33 millimeters per day. The resting phase, the hair loses the attachment to the follicle and becomes what we call a club hair. The club hair is shed when the follicle is reactivated and new hair formation begins. The variations in growth rate and duration of the cycle result in different lengths of uncut hair. We can see it here. Our active phase, where the hair is growing, then we have a regression and transition, and it pulls away from the follicle into the resting phase, and then we have a follicle reactivation to replace the growth of that hair. Here we can see an apocrine sweat gland. Apocrine sweat glands are found in the axillary region of the armpit, as well as the groin and in the nipple. They produce a thick, potentially odorous fluid. They mature primarily during puberty and produce a much thicker substance. It's got a cloudy secretion. It sometimes encourages bacterial growth, which increases the odor. It's the source of pheromones in many mammals, but this is a debatable thing in humans. The rest of the body tends to have merocrine sweat glands. These are the most numerous and widely distributed. They usually only extend into the reticular layer of the dermis, and they discharge sweat, which is a combination of water as well as sodium chloride. And this is via the sweat ducts that go directly onto the skin. And the function of sweat is for thermoregulation and general excretion or elimination of electrolytes, and then of course for protection, which helps to inhibit bacterial growth. Nails are found on the dorsal surface of the fingers and toes. The landmarks of the nail structure are as follows. There's the nail body, which is the visible portion of the nail that we see, and it's thick sheets of keratinized epidermal cells. Then there's the nail root, which is epidermal fold where the nail production occurs. Here we can see the nail, including the nail root and the nail body. Nail is going to be, again, an overall compaction of keratinized cells similar to our hair. As you get older, several things happen to the skin. The epidermis will thin, meaning the germinative cell activity is going to slow down, so the entire layer will thin out. You'll have fewer Langerhans cells, so you'll have poor overall immune function, and the skin will lose some of its ability to heal. It'll also have a reduced vitamin D production. Skin typically becomes a little bit lighter in color. A reduced melanocyte activity, which means you're more susceptible to UV damage, and a loss of secondary sexual characteristics. Usually there's changes in body hair and body fat distribution for both men and women, and hair follicle activity slows or ceases. The skin will become thinner overall, and will have a reduction in skin blood supply, and the activity of the sweat glands will stop as well. Sometimes we cut ourselves or damage the integument, and luckily for us, the integument is capable of extensive repairs, although there are limits to this in the cases of severe burns, deep and long cuts, etc. Now, this repair process occurs entirely locally with no input from either the nervous system or endocrine system. That is, the repair is under local control from the skin itself. There are 4 phases to skin regeneration following the injury. The first is the inflammatory phase. Immediately upon injury, mast cells in the papillary layer trigger an inflammatory response. This is a programmed natural response that attracts white blood cells to the region and fights invasive organisms. It also initiates healing and clotting of the ruptured vessels. Just a reminder that inflammation itself is where there is heat at the area, reddening, and an overall increase in swelling. The second phase is the migratory phase. After several hours, a scab has formed over the area, and cells from the stratum basale migrate along the edges of the wound. White blood cells, or macrophages, are removing debris and pathogens. The germinative cells of the stratum basale divide and replace the lost epidermal cells, and if the wound is deep, further epidermal repair must wait until the dermis is actively repairing itself. The proliferative phase, 1 week following the injury, the scab is undermined by new epidermal cells that migrate over the meshwork of proteins created by fibroblasts, or stem cells, from the dermis. Scarring phase. After several weeks, the scab is shed and the epidermis is complete. A shallow depression usually marks the wound site, but it will be filled by cells from the dermis, which will elevate the epidermis to its normal appearance. In more severe wounds, severely damaged hair follicles, glands, or muscle are seldom repaired and are replaced with fibrous scar tissue. Here we can see some sample pictures of some of the initial phases. Note on the left, the clotting occurs immediately following a wound to where we have inflammatory chemicals that are released. This would be an example of the inflammatory phase moving into the migratory phase. The second step here, epithelial cells multiply and fill in over the granular tissue. This is the migratory into the proliferative stage where they start to repair into that area. As many blood vessels as possible typically enter into the area to release macrophages. So it's a general process of cleaning up debris and repairing the skin tissue. And finally, restored epithelium will thicken and the area matures and contracts. So this would be a combination of proliferative and scarring phase in this last picture. And that makes up Lecture 8.